Let's continue our discussion about practical approach to thyroid FNA. In our last video, we talked about Dr. DeMay's two handy rules of thumb for interpreting thyroid FNAs. The first is that, since over 85% of thyroid nodules are benign, we should assume a thyroid FNA is benign until proven otherwise. The second is that, because more than 80% of thyroid cancers are papillary thyroid carcinoma, PTC, thus a thyroid FNA is either benign or PTC. These two approximations help simplify and streamline our diagnostic approach. So, when you're looking at a thyroid FNA, the first question to ask is this nodule benign? A benign diagnosis relies on identifying benign follicular epithelium. These cells usually appear in a nice, clean honeycomb pattern, like what you see in these two images. The nuclei should show minimal overlapping or crowding. They're small, about the size of a red blood cell or a small lymphocyte nucleus. They're round, have smooth nuclear membranes, and show dark, evenly distributed chromatin. In short, small, round, and dark. That's what benign nuclei look like. But if the nuclei aren't small, round, and dark, it's time to move on to the next question. Could this be papillary thyroid carcinoma? The good news is that PTC can be accurately diagnosed on cytology, because its nuclear features are distinctive and easy to spot once you know what to look for. Let's take a closer look at those features. In this quick stained image of PTC, notice that the nuclei are neither small nor round. Compared to the red blood cells in the background, the nuclei are at least 4 times larger. They're mostly oval in shape, and the honeycomb architecture is gone. There's clear nuclear overlapping. Here's another image from the same case. Again, the honeycomb pattern is lost. There's nuclear crowding and overlap, and the nuclei are clearly oval and enlarged compared to background RBCs. Now take a look at this pap-stained image. You can see that the nuclei are quite large. Compare them to the nearby small lymphocytes. These are highlighted in red circles. The follicular epithelial nuclei are oval, not round, and the chromatin is pale, not dark. You can also see small, distinct nuclei. We can also see that the neat, orderly honeycomb pattern is gone. Instead, there are areas where the nuclei are tightly packed, showing significant overcrowding. There are also areas with empty space. You could easily fit several nuclei in. Now, not every case of papillary thyroid carcinoma looks exactly the same, but they all tend to show a constellation of characteristic nuclear features. In this example, you can clearly see nuclear overcrowding, pale chromatin, and irregular, thickened nuclear membranes. These features alone are enough to confidently diagnose PTC. Studies have shown that PTC nuclei actually lose their usual structural rigidity, they become abnormally pliable. And thanks to 3D reconstructions like the one shown here, we can actually visualize how PTC nuclei fold and bend into odd shapes. This explains why we see things like nuclear membrane irregularities, grooves, bizarre nuclear shapes, and intranuclear cytoplasmic inclusions under the microscope. Here's a good example. You can clearly spot nuclear membrane irregularity, weird nuclear contour, and well-formed intranuclear cytoplasmic inclusion. These mirror what we see in the 3D models. This is another image. We see more classic PTC features, pale chromatin, irregular nuclear membranes, small but prominent nucleoli, and nuclear inclusions. These are all strong indicators of PTC. Let's talk a bit more about intranuclear cytoplasmic inclusions, since they're such an important feature in thyroid cytopathology. According to Dr. DeMay, when you see a true INCI, there's a 99% chance the lesion is malignant, and 99% of those are PTC. So being able to recognize a true inclusion is essential. Here's how you know it's real. It takes up at least 1/3 of the nuclear area. It's sharply defined. And it's the same color as the cytoplasm. Here's a textbook example of a true INCI. Note the size, the crisp borders, and the matching color. And here's what not to call an INCI. This one's too small and not clearly defined. Over here, we're looking at a bubble artifact. It might be sharply defined, but it's too small and not the same color as the cytoplasm. Now let's wrap up by summarizing the key nuclear features of PTC we've talked about. Nuclear enlargement, usually 2 to 3 times larger than a red blood cell or a small lymphocyte nucleus. Features related to abnormal nuclear pliability, including intranuclear cytoplasmic inclusions, irregular nuclear membranes, nuclear grooves, chromatin, fine and pale, often accompanied by a thickened nuclear membrane, and small, prominent nucleoli. All of these features are readily visible in cytology, which is why PTC can be diagnosed so accurately based on cytologic findings alone. Now that we've covered the nuclear features of papillary thyroid carcinoma, let's take a look at its architectural features. First up is the papillary structure. As the name suggests, papillary thyroid carcinoma tends to grow in a papillary pattern. In this image, we see a papillary structure without a fibrovascular core. But in other cases, like the picture on the left, you'll find papillary structures with a fibrovascular core. Sometimes, only the tip of the papilla gets sampled during the FNA. In those cases, you might see a tissue fragment with a smooth, rounded outline, like in these two images. That's still a papillary structure, and we refer to it as a papillary cap. Another architectural feature to look out for is the monolayer branching sheet. These are large, flat tissue fragments that get torn during processing. They can stand out under low power and are characteristic of PTC. Now let's talk about a rare but very specific feature, cellular swirls. These show up in less than 20% of PTC FNAs, but when you see them, they're highly specific for PTC. One study even showed that cellular swirls don't occur in benign or non-PTC cases at all. Morphologically, they look like circular whorls of overcrowded follicular cells, kind of like a swirl of tangled nuclei. You can usually spot them at low magnification, and they're a very helpful clue when present. So to summarize the architectural features of PTC. Papillary structures, with or without fibrovascular cores. Papillary caps. Monolayer branching sheets. Cellular swirls. That said, it's important to remember, the diagnosis of PTC is based on nuclear features. Architecture can support the diagnosis, but it's not enough on its own. You can absolutely make a diagnosis of PTC based on nuclear features even if no architectural features are present. There are also some other helpful features of PTC to be aware of. Psammoma bodies. These are layered calcifications, see the image on the left. In the thyroid, their presence is highly suggestive of PTC and reflects papillary growth. Bubblegum colloid. This is the thick, stretchy colloid often seen in PTC, shown in the image on the right. It's distinct from the watery colloid we usually see in benign nodules. Multinucleated giant cells. These are bizarre-looking large cells with multiple, sometimes more than 200 nuclei. Psammoma body, bubblegum colloid, and bizarre giant cells are just soft signs of PTC. None of these are specific enough to make the call on their own. So here's the bottom line, PTC can be accurately diagnosed on cytology, and that diagnosis rests on nuclear features, including nuclear enlargement, irregular nuclear membranes, nuclear grooves, pale chromatin, small, prominent nucleoli. Architectural features, like papillary structures and cellular swirls, can help, but they're not required, and not sufficient by themselves. And that wraps up our review of the cytologic diagnosis of papillary thyroid carcinoma. In our next video, we'll shift gears and talk about the cytologic diagnosis of follicular neoplasm.